Marta, we can start. So good morning, good afternoon, or good evening. We are very happy to have Professor Dominik Bucher from Germany as a presenter on NMR microscopy using quantum defects in diamond. And let me briefly outline Professor Bucher's academic career, which started long ago, but most prominent milestones are his master's degree in 2010 in chemistry from Technical University of Munich. Then he went to complete his PhD in biophysics at Ludwig Maximilian University, Germany, where he utilized ultrafast spectroscopy to study UV-induced damage processes in DNA. With a growing interest in potential applications of quantum technology in chemistry, Dominik moved to Ronald Wolfsworth's group at Harvard for his postdoctoral work. In the spring 2019, he started his independent career as a junior research group leader at Technical University of Munich. And in 2022, he was appointed as Rudolf Nussbauer Professor. Dominik uses defects in diamond known as NV centers as quantum sensors for NMR spectroscopy in the nano and microscale range. His research group works at the unique intersection of quantum sensing and chemistry, utilizing interdisciplinary methods from applied quantum physics, material science, and biophysics. The goal of the research is to apply NMR spectroscopy at the smallest length scales possible, from nanoscience and surfaces to microfluidics and single-cell biology. Dominik, we are all here to listen to your presentation.
Olga, thanks. Of course, first of all, for inviting, but also for this very detailed introduction. And I think you gave a perfect overview also what my talk will be. So I will talk about using NV centers in diamond for performing NMR spectroscopy on the small length scale, but also NMR spectroscopy with imaging capabilities. Okay, let me just line this here. I hope you can see everything here. So let's start. So I will start not from the diamond side, but more from the application side. That's what we would like to do in the long term. So I think most of you know MRI, from hospitals and medicine applications, which helps us to gain understanding of our bodies and brains, such as here. A method which is more specialized, but I would say similarly important, in particular for chemistry, is like the sister technique, which is NMR, so nuclear magnetic resonance. It relies on the same principles like MRI, but what we do here is we do spectroscopy, and these techniques allows us to perform or to analyze molecular structures in very high detail. So you see, for example, here this ethanol molecule, and looking at the NMR spectrum, which basically gives information on the precision or the Larmor precision of each of these nuclei, the protons here. We get this very detailed spectrum, which then gives us the information about the three-dimensional structure. So I always say it's like the eye of the chemist. So these techniques are very powerful, but there are some limitations. So let's start with how normally NMR is done. So we bring our sample into a magnetic field. If the sample has nuclear spins, so typically it's protons, could be also carbon-13 or fluorine, these spins will start to precess in the external magnetic field. And this precession of these spins comes, of course, with a precession of magnetic field, which is then typically detected with a coil, which is then read out by a computer. And as you might see here, these coils, of course, can be miniaturized. So if you're interested in smaller and smaller samples, but at some point you will hit a limit. The first question, of course, why do you want to look at small samples? So typically NMR needs kind of these 200 to 300 of microliters of volume in these NMR tubes. If you want, to go to single cells or maybe even single molecules, this induction technology does not work anymore. especially if we go like sub 10 micrometers. And that's also related to the MRI. It has, of course, a little different constraints in resolution and volume, but overall, it's very similar here, that we lack kind of a detector for very small length scales. And the solution is, well, replacing the detector from our classical inductive detection with our solid state spin system in diamond to detect these NMR signals. So the vision we have is to build or to design or have like a tool which is like a hybrid between the MRI device and an optical microscope, which can basically zoom in and detect NMR signals on the small length scales, where of course the heart of the technology is then a nitrogen vacancy center doped diamond. And to give you an overview, what I will talk about in this presentation here. So I will show you that the nitrogen vacancy center is a solid state spin defecting diamond. I think most of the audience will know that. It can be used as an atomic sized quantum sensor, in particular for magnetic fields. That's what we are interested in. And it's a novel tool to perform a new kind of spectroscopy, which merges optical and magnetic resonance spectroscopies to perform microscopy and spectroscopy. And this is a tool where we think on the long term, because we have to develop this technique into these directions for lab-on-a-chip applications, for single cell studies. You can think about probing tissue slices, but also material science. Okay, so let's start with a quick tutorial on the quantum sensing side. So how does this novel NMR or MRI detector work? I think in this audience, I have seen the previous speakers, so I think most people know what the NV Center is. Let me just very briefly remind you and bring everyone on the same page. So, we start with a host material, which is diamond, this beautiful, carbon-based lattice. And what we have is a nitrogen, which is shown orange here, and a vacancy. That's, the defect, what we will use as a sensor. And the question is, why is this defect in the diamond so important or so useful for us? Well, because it has three distinct properties. So first of all, we can optically polarize its spin state. So this defect here, you can think about as a electronic spin is sitting in the diamond lattice. So we can optically polarize the spin state. We have very long coherence time of this electronic spin because it is kind of confined or protected from the environment by the diamond lattice. And we can coherent manipulation at room temperature with microwave pulses. And very important, we can also use optics to read out the spin state of this defect, because depending on how the quantum state, the spin state of this defect is, we have a different intensity in fluorescence. Okay. And you see these three criteria define a fully functional qubit, again, importantly at room temperature, which is of course also important if we think about biological applications. Okay, so that's how it looks like in our lab. So I have to disappoint you. We don't work on nano diamonds. We have this millimeter-sized diamond chips. You see it in here. This is like a 2 by 2 millimeter diamond chip. This diamond is doped basically everywhere within NV Center. So you see here going through the laser, which is green, and now we just filtered here the green light. So you see basically the red emission, which is the fluorescence, which is emitted by the NV centers. And I told you before, the emission is spin state dependent, which we then will use to read out our quantum sensor. So how does it look in our lab? So let's see if I can start this video. So that's like a typical experiment setup we have in our lab. As you see here, you see a lot of green light. And I will just show you here the red fluorescence of our NV diamond chip. Let's see if this works. Doesn't work, huh? Ah, now it works. So you see here basically this 2 by 2 millimeter diamond setting, and you see the optics around. Now, if we use a laser goggles, which kind of blocks the green light, we see the very highly fluorescent NV diamond. Again, what we used here is a very highly doped diamond to visualize the effect much more. And what you see more here's like the microwave transmittance going on the diamond, and on the bottom we have some optics to collect the fluorescence light. And so how the experiment works is that, as I told you before, this fluorescence is spin state dependent. As you also know, the spin couples to magnetic field. So what happens now is that we use these defects in diamond for translating magnetic field into optical signals. This is shown in the next video. Oops, here. So what we have here now, we have now connected the fluorescence detection, it's an APD, it's an average photodiode, to an oscilloscope where we basically can read out here the photo voltage of this APD here. And that's exactly, I told you before, that's what we read out at the end. So we read out the spin state with the fluorescence intensity. So if something happens here, we know that the fluorescence of the red light is changing. And what does the student do here? He has a magnet in his hand and basically moves up and down the magnet. And as you will see by changing the magnetic field locally, by moving the magnet forth and back, we see that the fluorescence intensity is changing basically. And this is basically the most fundamental principle that we now can translate the magnetic field here into an optical signal. Okay, so oops. So now let's move on and let's discuss a little bit more from the physics side how this exactly works. So I didn't talk much before about how exactly the photophysics work. The only thing you have to remember here is that we have spin sublevels in the ground state. It's a spin triplet. And that the spin ms0 with the sublevel ms0 is a bright and the ms plus minus 1 states are dim. Okay, so typically now we have it's not 100% on and off, but we have 70 or 100% on and the dimmer state here. And what we typically do is we use then microwaves to address two of these levels, and then we have a two-level system which we basically can represent here on a Bloch sphere. Where then the north pole is our bright state, so the ms0 state, and the south pole would be here, the dimmer states, for example, in this case, the -1 state. Okay, so what is the general idea of this quantum sensing? Well, we typically start with a specific quantum state, which is typically a equal superposition of spin up and spin down. Okay, and this superposition, as we know, is very fragile. Typically will decohere. And when we always talk about the quantum technologies, typically if you do quantum technologies, quantum information, quantum computation, you would like to protect these decoherence. So you would or avoid decoherence. What we in quantum sensing do is we are kind of in favor of this interaction with the environment. So the environment interacts with our qubit. There will be decoherence or in this specific case here, we have some phase accumulation, and this is what we take to measure. So we watch basically the impact of external perturbation on the qubit, which we then at the end can read out because we know that the different spin eigenstates have different fluorescence intensity, and then of course we can start the experiment again. And this interaction can be different things. It can be temperature, it can be strain within the diamond lattice, it can be electric fields. But as I said before, we have a spin state here. So the most obvious interaction are magnetic fields. And they can be, that's also the important point for the V center. We can have, very different modalities of sensing. It can be static magnetic fields. It can be NMR signals, so nuclear magnetic resonance signal. It can be electron resonances, but it can be also just a magnetic noise, induced by materials, for example, which we can sense with this kind of technology here. So as I said, the main goal of my lab is to apply this technique for magnetic resonance techniques. So how would this work? So let's go a little bit into more detail. So we have our diamond. We have here a single NV center. And the single NV center, of course, behaves like an electronic spin sitting in this, again, protected environment of the diamond. And if we apply some magnetic field B0, what will happen? I'll begin to change. It will start to process at its Larmor frequency. Okay, that's just very basic physics. And what we typically do in the magnetic resonance community, we kind of don't watch this Larmor precession from the laboratory frame, but we basically change the coordinate system so that the frame is rotating at the Larmor frequency. Oops. That's then the rotating frame. So now we see the frame is rotating at the Larmor frequency, but then the spin state stays static. Okay, just a coordinate transformation, what we do here. Okay, so far nothing special happened, but now if we bring another molecule, or let's say any other spin nearby, or magnetic material, what will happen? In this case, we just go back to our ethanol molecule I showed you on the very first slide. These protons, of course, have magnetic moments, which will basically have some impact on the NV center. And you can see here, we can just draw these dipolar lines here. But of course, that's what it is. It's just coupling, dipolar coupling in this case. And what we will do is we have not only omega 0, which is the pure Lama frequency at our B0 field, but on top, of course, we will have now the magnetic field induced by the coupling. So we have omega 0 and omega s. In the laboratory frame, that would mean that you basically now with this video, we won't see any change because typically, of course, the coupling is much weaker than your B0 field. But in the rotating frame, the rotation which occurs, or the precession which occurs now is exactly at the omega s, which is exactly now this interaction between these two spins here, or between the NV and the molecule spins here. Okay, and again, you see like this, again, we get this what we call phase accumulation, which is, gives information about the coupling. And this we can then basically, so it here, it's basically, omega t, that's how far the spin vector of the NV center has moved. And then, we do some more rotations on the Bloch sphere so that we can at the end read out this quantum state again by the spin-dependent fluorescence. This is, again, a very, simplified picture, how the sensing works at the end in detail. And we have to discuss, case by case, because I told you before, we have very different options of sensing, and then of course also depending on the pulse sequence, different capabilities. Okay, so now the question, of course, is how far do we see? So if we bring the NV center to the surface, so we are, as I said, mainly interested in sensing something outside of the diamond. What's the detection volume? And what you do is you would basically, if you go back to the previous slide, you would kind of integrate over these dipolar moments here. Let me go back to the laser pointers. You would integrate, if many molecules, you would integrate over these dipolar moments, and then you get this kind of roughly rough rule here that the detection volume with a hemisphere with a radius equals the depth of the NV center. So if the NV center in this case is a couple of nanometer deep, then your detection radius would be also a couple of nanometer. And this scale, so if you bring NV center deeper and deeper, then the detection volume gets larger, but typically the signal, depending on how you what you measure, the signal, of course, in some cases goes down. Okay, and we distinguish now in single nanoscale NV NMR. The next one is what we mainly do in my lab is nanoscale ensemble NV NMR. So we take many NV centers which are close to the surface. So that's for it's again nanoscale because each of these NV center has its nanoscale detection volume. And then the third one, which we kind of categorize, is then a kind of couple of micrometer thick layer, and then the same rule roughly applies. We go then to this kind of micrometer volume. And what is the advantage? You might see here that's only a disadvantage to go from a single NV to an ensemble because, of course, you lose this kind of very beautiful nanoscale spatial resolution. That's true, but at the same time, of course, you win sensitivity because you average over many NV centers, and this makes you, gives you higher sensitivity, but also gives you more robustness because, of course, you have a much stronger fluorescence, and which is good in particular if you do chemistry on the surface. What are the applications? Of course, here still, I think the main goal of the community to think, what can we do at some point, single molecule NMR. As I said, my lab is at the moment for technology developing, focusing on ensembles because they have higher sensitivity and easier to handle. And also we see here a couple of key applications, such as in this case here for material and surface science. There are many open questions where you would like to address interfaces and surfaces. We think that the NV center is a good tool for that. And at the same time, if we scale up the volume, we can get here to maybe 10 micrometer, and you see 10 micrometers the length scale of a cell. So here we think that we can do life science studies and lab on a chip applications. This also brings me to the overall look of my group. So we kind of divide the group in three parts. One thing is this nanoscale magnetic resonance. I told you before, mainly for material science, where we look at interfaces and so on, and application and catalysis, thin film materials, 2D materials. We won't talk today about that. We will mainly talk about the microscale magnetic resonance, where we at the moment not yet there to do biology, but we are preparing for that we're developing technologies for performing magnetic resonance in microfluidics, that we look at diffusion of water. We will talk about this, and also our newest results here, where we have basically developed an optical microscope which can perform magnetic resonance. And again, the applications are here, ranging from single cell studies to lab on a chip, and also high throughput analysis. But of course, could be also used here for material science. And at the end, the main work we are doing is on the technology development side. I said also this is, of course, strongly connected here, where we, of course, look at, how can we improve diamond materials, and where we talk about or like discuss novel sensing schemes for sensing specific frequencies, improving sensitivities, but also looking at some of the competitors of the NV diamond, such as novel spin defects, for example, in hexagonal boron nitride. And of course, we was interested then in forming startups and to perform technology transfer here. Okay, so let's start now with the science, and let's go a little bit through these microscale NV NMR applications. So the first thing we needed after the proof of concept that we can perform with NV centers NMR spectroscopy, we need like a platform to handle the sample. And what we developed here, and was mainly done by Robin Allard here, that we have a diamond. So the diamond is sitting down here. It's a trapezoidal shape of the diamond. Maybe let me show you the next slide. So here we look on the top. So we take an image exactly from perpendicular to the top. The diamond has this trapezoidal shape because we couple in the light in a specific way through these polished edges. That's not so important for now, but this avoids basically having laser light on the sample, which is of course important for biology. And then this diamond is glued in like in a pocket of this glass chip, where on top we have here this microfluidic channel here. So the bottom is formed by the diamond, the top is formed by the microfluidic chip. And now the important thing is that we can have basically any arbitrary channel structure on the diamond. So you see this is now the diamond surface, which is heavily doped with NV centers. So the bottom is basically our sensor. And what we can do now in here is we can put our microwave loop on the top and can move our laser spot around and perform here primitive NMR spectra. And I guess the question will come later, what's about the resolution here, because I told you in the very first slide that the power of NMR spectroscopy is to get very narrow lines, because the narrow lines are the more information about the structure you get. And here with this technology on the microscopic length scale, you see this is like 80 micrometer in length, and this channel here. We are mainly limited by our own engineering capabilities. So here you see the line width is a few hertz, and this is mainly limited by the magnet we are using at the moment. And we're still working here at low field. This is the main reason for that. It's much easier. We can talk later about this in the discussion. But here this is done at 0.2 Tesla at the moment. Okay, so that means basically now we can perform NMR spectroscopy on a lab on a chip device. Again, it's primitive, but it's I think an important step for future applications. And one first thing was, how can we do now? What can we do now? We can do molecular analysis, very primitive one at the moment. I show you here, but at the same time, NMR can do much more than only doing spectroscopy. For example, it can be also used to detect diffusion. And this is an important topic. Just to motivate here is that people understand diffusion very well if you think about single molecules or atoms. And it's used on a daily basis, again, if we come back to our MRI device at the beginning. So if you go to the hospital and get a brain scan, there's different ways of analyzing and performing the data. One thing is to measure the local diffusion coefficient of the water in brain, which gives you information about the underlying structure, and you can see here. And the good thing is people learned how to interpret these results, but there's a lack of fundamental understanding where this kind of differences in diffusion come from. Obviously, we have different compartments, and of course, water at some point will hit a wall. It's now seen down here, for example, if the water diffuses around in a channel here and hits a wall, of course, it cannot diffuse beyond this wall. And this basically gives you different apparent diffusion coefficients. For example, in this way, it would be not limited, and this way would be limited. The same thing happens exactly on this meso scale, on the single cell level in your brain. And this gives then you at the end this diffusion weighted MRI images again, but a real fundamental connection cannot be done because there's no technique available which really can zoom in and measure local these diffusion coefficients. And you may see here, or may recognize here again the structure. This is exactly what I showed you before, our microfluidic structure. It's also shown here. The NV center, or our NV magnetic resonance spectroscopy setup, in principle is the right tool for doing that, because we can exactly measure locally resolved NMR signals. So how is this done? Maybe just a very brief reminder. Probably most people are not that familiar with NMR, but we'll just give a very quick overview of how we would measure diffusion with magnetic resonance. So what we typically do is you have polarized your nuclear spins, and you apply these pi half piles. Again, very similar with the NV centers. So people who are familiar with NV centers, the same thing, of course, we do in NMR here. So all the spins you see that's in the space, that's X, Y, Z, will be like, aligned. So they all get tipped down the gravitatory plane. And then you will basically apply a gradient. So that's the most important point. We might apply a gradient. So that basically means the Lama precession changes over space. And what that means basically, it will basically kind of lead to this kind of dephasing of our spins depending on the position in space. Okay. And now what we do, we can apply a pi pulse, so like in a spin echo, and have a second refocusing gradient. Okay. So let's just go through. So if we have basically no molecular displacement, so let's assume we take a solid sample. What will happen these spins, as I told you, will be rephased by this magnetic field gradient. You add your minus sign of the pi pulse here. And what happens, what we know from our spin echo, it will basically kind of rephase, and we will get then here a with the second gradient, we'll get here then our spin echo, where basically these induced gradients will disappear. So what happens now if we have diffusion? And now that's exactly what we want to go into. So if we have now, of course, these spins change their location here, so they diffuse along this gradient, you will not be able to refocus all of the spins, and this will lead basically to a reduced signal here in the spin echo. And this we can use to perform very precise measurements of diffusion with magnetic resonance. Again, I think it's a very beautiful example of how powerful NMR is. You cannot do only spectroscopy, but for example, there are many other of course situations, but you can also measure here diffusion in a very beautiful way, I think. So what we need now for the NVs to implement this, so we need magnetic field gradients, and this was, we thought it's easy to do. At the end, we needed some help by Maxim Zaitsev. He's an expert on developing gradients for MRI devices. So he developed us these gradient coils here. So we have a gradient across our microfluidic structure here. And the good thing is with the NV center, of course, we can directly image and, basically, visualize the local magnetic field. And you see here the gradients which are induced by these coils can be in X, Y, and Z direction. And now I don't want to talk too much about the pulse sequence. So we exactly do this spin or gradient spin echo. So we apply now this spin echo with these pulse gradients. And with this, we can calculate back the diffusion coefficient. And since we can do this in all 3 dimensions, so in X, Y, and Z, we get these ellipsoids here. Basically, it shows you here, for example, let's go to the narrow channel here. We have limited diffusion confinements on high diffusion coefficient in the Z direction, but a limited one in the X direction. And similar here, it's less confined in the X direction, but more confined in the Z and the Y direction. So, and this, of course, you see we can do for the different spots. You see it here in the 3-dimensional a little bit more. Of course, there's some deviations. It's experimental data, but this is now a technique where we can, spatially resolved, make, specific measurements at specific locations. And again, that's, of course, only a proof of concept. We did not do anything new in here. This is all known before, but we really think that this technology paves the way for many cool applications in future. So as I said, the main motivation came here from really this magnetic resonance imaging validation. So could we now, for example, really have like, specific phantoms, or maybe at some point really like, tissue slices to measure the local magnetic field gradient, overlay them with optical images, and then basically understand more the, the diffusion on, on a single cell or like on a micro scale in tissue. Another advantage, of course, is since everything is miniaturized, we have 10,000, we could go up to 10,000 Tesla per meter. That's an insane value, but since everything is miniaturized, you can make your coil small. That's possible. So you could do really here diffusion measurements on extreme scales. As I said, with this very high gradient, you could, for example, allow for very small diffusion length, which cannot be accessed before. So you could also really access like a new parameter space with these microscale and techniques, because you miniaturize everything. And of course, will be less important for this audience here, but could be also, of course, for material science, where ion diffusion plays an important role. Okay, so this is our diffusion MRI device. And of course, you can ask now, we did here like a scanning approach. So we moved around our laser spot, we measured here, then we measure here, and we measure here. And of course, if you think about optics, the question is, why can't you just detect everything on a camera? Because you could do then, in contrast to the scanning approach, you could do widefield approach, and you would get all the information at the same time. in particular for NMR, where all of these measurements, take a while. So that's exactly what we did, developed. So can we, as I said, this was the motivation at the first slide, can we build like a fusion or hybrid of an optical microscope and an NMR or MRI device? And so how would this work? Well, it sounds pretty easy at the first place. Again, you take our microfluidic channel, our diamond. You see now this trapezoidal shape where we go in with a laser bouncing off and kind of avoids laser illumination of the sample. And then, we would just image everything here on the camera. So before we just collected all the light. Now we would image this on a camera. So you may say, oh, that's kind of trivial. At the end, it's not, because what we have to do is we have kind of, get this NMR signal, which is kind of, this free induction decay, what is then, translated by the NV centers into magnetic signal. And we have to make a movie kind of this, free induction decay. So what happens is kind of this NV center here translates this, again, this NMR signal into an optical signal, which is then basically oscillating in the time domain. And that's very distinct from the previous quantum diamond microscopes, where typically where you don't have to get time resolved data. But if you do this NMR detection, what we do here with our approach, you have to take a movie of your NMR signal over time. So what you need here is a high-speed camera to take very fast images. And then you see, over the time trace, you will get basically then your, free induction decay for each of these pixels. And then you can take the Fourier transformation like a normal NMR to take your spectra. And as said, it was not a trivial task. Apart from, getting a good camera for this purpose, is also that everything here needs to be absolutely non-magnetic, because NMR signals, of course, are very fragile. Again, everything which is magnetic will impact your NMR signals. So you're getting like optics, objectives, which are absolutely non-magnetic, was a long way to go. And just now see the big advantage of this technique. So first of all, the imaging happens in real space. So we do imaging in real space, which is very different if you would do MRI. And what we discussed before here, you typically use gradients similar to the diffusion experiments, but you use gradients to encode the information in the space. So you apply gradients. So at the end, you go to the K space, whereas here we can really do MRI in the real space. And most importantly, is, if we, in this case, we had a 1.1 million megapixel camera, we get 1.1 million NMR spectra at the same time. And we see, of course, at the end, lots of data. So let's look a little bit more detail how this works. So we have, again, our microfluidic chip here. Now it's a bit more complicated shape, what I showed you before. And we just zoom into this area here, where we now basically image here our NMR signal. So how does this exactly work? So let's just go to show this here. So each of these points here is basically an NMR spectra. So in this NMR spectra, keep in mind, again, comes from a time trace of images, where we then at the end take the Fourier transformation and then have the NMR spectra, and then we plot here only basically the signal to noise of this image here, okay? And you see clearly how the signal, the SNR, gets better and worse again. This is exactly where our water signal in our microfluidic channel is. Okay. And just, there's of course other different spots, the red one and the black one and the blue one. And we could do different geometries. So we have imaged different geometries. Again, the most important point for NMR is you can only look at the water intensity and the signal intensity, but also at the phase of the NMR signal, where you see some interesting geometry effects, but also the frequency and also the line width, which gives you information about local magnetic field gradients, and of course in the long term also to see here really spectra to do chemical analysis in a highly parallelized way. So where does this idea go? So I think one thing you see here is a diamond chip. The cell is grown on the diamond, basically. One idea is to do perform single cell studies. Of course, I have to also mention here that's not that easy. We can do this today or tomorrow, and we of course have to improve the sensitivity a lot to really see signals from single cells, because you can imagine that of course metabolites, but also other signals in cells are very weak. Another approach, of course, is that we mentioned before, that you can do this highly parallelized NMR spectroscopy for high throughput applications. But similarly, of course, also think about NMR microscopy of thin film materials and so on. So I would say the application space is basically infinite, but really we have to work on the technology to improve sensitivity, which is always a main limitation of NMR. But then it really opens up a totally new way of performing microscopy. And now, I'm not sure how much time I have left. I forgot to put a do we still have time for one more topic?
Dominik, please go ahead.
I go ahead. Okay,
Please,
good.
yeah. No limitations. Yeah.
Without limit, I shouldn't have said this. Okay, then I move on. So the next part is a little bit more technical. I will come later back to the cell biology again, but I would just to show you like a second way of performing imaging. This might be in particular interesting for, again, people working with NV centers and diamond, but I would also say this might be a very nice technique also for people who use nanodiamonds. So it's a very general and simple technique, which I think is very powerful. So let's go through what I mean with this new readout, what we call time to space encoded readout. So if you have the NV center I showed you before, you see it here, we have the optical excitation, you have the microwave pulses here. And typically we have our pulse sequence, we have the readout, we initialize the spin system and start again. This is all done basically, see down here. So what we do is, we manipulate the spin with microwaves, and then we basically read out and re-initialize the quantum state, okay? And of course, if you would do now imaging, you would go to the next spot and repeat everything again, apply the microwave pulse sequence, to the readout and then initialization. And now it's a very simple, but I think powerful idea was that why not just applying the pulse sequence? Of course, keep in mind, typically the pulse sequence is much longer than, your optical readout initialization. So why not doing the pulse sequence on an entire, volume, or let's say region of interest? So we would, manipulate all of these spins. What we of course would also do in this case, typically we don't apply the microwave only on one spin. So typically would do it like on a, let's say, a couple of hundreds of micrometer. And then what we do is, then basically we read out. So we manipulate all of the spins, and then we read out these NV centers very quickly, one after the other at different positions. Okay. So what we do is, we manipulate all with the microwave, and then basically, we scan here, read out here the quantum state, here, here, here. And of course, this has to happen much, much faster, or very fast in order to, of course, don't lose time between you scan your laser spot here. And you may say, oh, that's a very complicated thing. How would I do that? And the nice thing is basically all pulsed NV experiments can do that, because we can use just our AOM, so our acousto-optical modulator, which are built in an experiment. So we use typically the AOM for kind of turning on or pulsing our laser. But at the same time, we can just feed in different frequencies, and this basically leads to a deflection of the laser beam. And this basically happens, at least on the time scale I'm talking about, on the real time scale. So this can happen like on a few tens of nanoseconds. So we can move very quickly the laser spot from one position to the next one. In this case, if we have only two readouts, by just putting in two frequencies in. And we've done this really, I think, when we had this idea, this can be done within one afternoon. You can just upgrade your experiment by just using the infrastructure you have, and maybe just adding like two frequencies to your AOM. And so what can we do with this technique? And it has two main I think very powerful applications. One thing is, again, more for the sensing people. The second one will be more for the imaging people. Is of course we have a lot of noise. We always have to fight with noise, and in particular also common mode noise, which we have also learned can be also introduced, for example, the microwave. There's a very nice publication by Victor Costa, where phase noise of our signal sources has an impact. And now the important point is, what we would do here is we have like a sample, and we have like a reference. This can be just done by covering half of our diamond, but of course there might be many other reasons or like other possibilities to do that. So we basically would manipulate both spins. So both spins experience the same pulse sequence, the same sensing scheme. So they have the same time they do the sensing. So all of the noise which is encoded here is encoded in both positions, and then basically read them out after each other. So what we get now, we get like a signal, and we did this for NMR, but again, this could be any different technique you can imagine, where we have one reference and one sample spot. So we get the NMR spectrum here and here, and again, the measurement was done at the same time. We just do the reference at different times, at different spaces at the end. And we see here that nicely the noise is very correlated, and this is exactly the point that if you have limited by microwave noise, of course, you would encode this on both positions. And you can easily subtract this, and then you see you can reduce the noise, depending, of course, on the quality of your microwaves. We saw here a factor of 2 to 3 in improvement. even with a pretty good signal generator. Of course, if you have like different systems, this may have much larger impact for noise cancellation. And of course, the nice thing here is if you also correct for laser noise, basically you can remove most noise sources with this approach. And again, in a very simple way that you only have to upgrade your AOM with 2 frequencies. So this is the first application is for noise cancellation. The second one is for imaging. So what we would do here is you have like a long pulse sequence. Also if you, for example, measure T1, so if you think about nanodiamonds, you measure T1 times, so you have a very long pulse sequence, and then you read out 5 microseconds, and you have to start again. So what we can do now here, again, it's shown here at the case of NMR, but again, I think especially for T1 measurements, this might be very beneficial. You would apply your pulse sequence, on all the spins. You have a long measurement sequence for milliseconds, and then basically would read out all the spins very quickly after each other. And we've seen this here, we just do the NMR with 4 spots. You can do this, with many more spots, and we can do primitive imaging here. But the most important point is you see that you win now massively in time because you can basically get, many readouts. So if you keep the readout time on the same time like your pulse sequence, you can, win orders of magnitude. So if you think about a very long pulse sequence on the millisecond time scale, you can accelerate your scanning, by a factor of 400. Instead of repeating the pulse sequence, reading out one spot, repeating the pulse sequence, you would apply the pulse sequence on all spins and then do a consecutive time to space encoded readout. So really think that's like, again, mainly due to the simplicity, it's a big advantage for us. And you see also before I mentioned nanodiamonds, you could think, of course, about, having a different nanodiamonds and, which all measure T1. So you have to wait for a long time. And so you could multiplex and measure many nanodiamonds at the same time with this technology. Okay. And since Olga told me I can speak as long as I want, I will quickly go to the last topic. Again, back to the bio thing. So, I mentioned before, of course, one idea is, to go to the single cell level. And the key question is, how feasible is it at all? And as I said, the vision is here to at some point to have like a cell on the diamond chip where you can perform then, different studies such as we saw before. You can think about water diffusion, but at the same time also, metabolomics. And this, of course, metabolomics means you would measure like, metabolites in the cells. But as you can imagine, these concentrations are low, and where can we in principle go? I get often asked also by people, what is about, why would you measure single cells? And this might be maybe not clear to everyone, but there's a very important point if you measure the ensemble. So think about, you have like a cell ensemble, you would always measure ensembles with NMR. And you have like 2 cell types there, cell A and cell B. So if you could take the mean, you would get the average value. Okay. But you would totally basically miss the key information that you have 2 cell types which have very different concentrations. And so the mean gives you the wrong value. The same thing for non-synchronized dynamics. You would basically get basically no dynamic, although, for example, the cells could oscillate and have different signals. So it's very important in biology to look at the studies at single cells. So let's look here. So how feasible is it to do experiments? So down here, it's a complicated figure, so I apologize for that. But here we have like the sample volume in microliters. So you go from a prokaryotic cell to eukaryotic cell to, here the egg cell. And you see here the detection volume or the detection sensitivity nano mole per hertz. And here the concentration. That's a little bit complicated. So let's go through together. So if we would like, did our first experiments, which I did during my postdoc time with von Walsworth, we would end up here, like, having this volume of roughly 10 to minus 5 microliter. This is around, 10 picoliter. And we would have a concentration. We measured pure water here, and this, ends up like in this 100 molar of water, which would basically mean, we can detect pure water in a second. So we are far away, of course, from anything which would be like, metabolomics, which is marked here in green. So we really need the orders of magnitude of sensitivity in order to detect like millimolar or micromolar concentrations. So what can we do? There's of course lots of engineering we're working on there. So we can think about, getting better optics, get more light. There's maybe a better readout. There's this quantum enhanced protocols where you can, have better readouts of the NV centers. there's better diamond material, of course, and of course, increasing magnetic fields, because this will increase your signal. So if you would, implement all of these, we are working on this now for a couple of years, and you see we are not yet reached, these orders of magnitude. It's a lot of work to do. We would basically go down here. So that also means at the end, if we think about measuring water, for example, this would be possible, but having like an information about metabolites, it will be very hard, because reaching sensitivity on a single cell level will be kind of an issue. However, we have other tools which can help us. And this is very well known that, if we think about the polarization in NMR, so, maybe for the NV community again, it's very easy. We hyperpolarize our NV spins by the optical pulse. In NMR, basically, we had low fields, 10 Tesla, but maybe it's low fields, high fields, but, low room temperature and high fields. But even then, if you think about the Boltzmann factor, we will have a very low polarization. So typically, we have only a few spins polarized within like a million spins at, as I said here, 10 Tesla in room temperature. But there are techniques which we call hyperpolarization, where we basically now can increase this number up to a few percent or a few 10% in some cases even. And also this we have partly shown, like during my postdoc time. So we use the technique where we use para-hydrogen, so you can easily go down here. And of course, if you now look even in, this was just a first attempt, if you really work like and improve this technology, we think that we can go down here. In particular, so this was shown without the technology improvement. So this was just the blue and the red one. And now if you would add like these technology movements we just discussed before, you would go down here. And what we used here is a technique which is called dynamic nuclear polarization, where you basically cool down your sample to increase spin polarization. You add electrons and transfer them this electronic spin polarization on your nuclear spins. So dynamic nuclear polarization. Then we end up here in a regime where we can detect, metabolite concentration, which are interesting for biologists. So at the end, the answer will be here. We very likely or very sure will need additional technologies to improve our signal. So the NV alone doesn't solve this issue. So we need hyperpolarization techniques. This can be para-hydrogen-based methods, but can be also like dynamic nuclear polarization techniques in order to perform metabolomics on single cells. If you, of course, only look at water, then, of course, this could be impossible with techniques we have now. Okay, with this now, I'm at the end. So we'll just, I hope, I could convince you that the NV Center is a very nice tool to perform a novel kind of optical magnetic resonance microscopy techniques. It's promising, so that we're not yet there, but working on that, and that we can find application, for lab-on-a-chip applications, single cell studies. I showed just what is feasible here, and for tissue slices and material science. And of course, very important, it's a novel tool looking for applications. I'm always open, for new ideas, and please reach out if there are any thoughts. And of course, here's like a review we wrote 2 years ago to show like, the initial developments in this field. With this, of course, I would like to thank, of course, the students who did all of the work, and of course, our collaborators, as I said, for example, Maxim Zaitsev made the coil, but in particular also Peter Knittel, who makes the diamonds for us for these bio applications. And of course, the funding agencies, and again, for being here and to present my results. Thank you.
Dominik, thank you very, very much for your comprehensive lecture for past achievement and future possibilities. So we are open for questions. Please feel free just to unmute yourself and ask your question.
Hi, Olga.
Hi,
Dominik, thanks, very nice talk. So I have a question about this widefield NMR, because I think you didn't specify what type of diamond you used in terms of doping, thickness of the NV layer, and also to get that signal, did you do any polarization? I mean, how you get that signal? So how you, there are many things I missed out.
All very good questions, all very good questions. Let me go through step by step, right? So, of course, the key question you have to ask yourself, what's like the spatial resolution? So we call it like magnetic resolution, because the limit, the limit would be the optical diffraction limit. ideally, okay. So the next thing is, it's like very similar to the other QDM application. It's the NV layer thickness. And we took here like a 10 micrometer layer, which also, I don't have now the figure here. It also, we can see a little bit here, that also defines like the spatial resolution. So with a 10 micrometer layer, it just, again, we told you before, like the detection volume is like, half of the, well, the distance of this from the surface of your NV center determines the detection volume. If you have this like 10 micrometer thick layer, you roughly detect also 10 micrometer in the Z direction. This sets your spatial limit, right? So this is like a 10 micrometer layer. I think it's around 10 to 15 parts per million dope.
So this use radiation, I guess, electron radiation.
Radiation, exactly, radiation and the standard thing, right? Exactly.
Okay, I see.
So now I forgot your second.
And is it a radiation to this 10 micron depths? Because probably it's huge damage for high power nitrogen atoms required to implant at such.
No, this is grown. This is grown with nitrogen and then
grown. Okay,
grown, exactly. So overgrown and then basically radiate with electrons.
Okay, it's what, yeah.
And now I forgot the second question.
The second question is the polarization. I mean, have you to get this?
Oh, right. So that's exactly the point you see. That was exactly was before. Very careful to say, like, you may ask, oh, why don't you do like any bio stuff? Well, it's not that. I mean, I'm just honest here, I think, and it's not that easy because what we did actually, we used again our favorite Overhauser hyperpolarization. With Overhauser, just for the experts, and we or non-experts, we add like, tempo radicals and then transfer the polarization of the electrons on the nuclear spins, which gives us a factor of 200 in SNR. Exactly. We have also to admit this. So if you look at the paper, and the sensitivity is not very good. And this is for many reasons because we did not focus here at all. For example, also our camera was kind of, the sensor was, in its own noise. So we had only like 10%, of filling the wells basically. So the experiment was far away from any optimization, but still we are limited by the sensitivity.
When you say Overhauser, I mean, you are using like tampo kind of thing, or I mean, you just CW irradiation?
So basically what you do is, before you start with the pi half pulse of your, you irradiate, I think, like 200 milliseconds to transfer the polarization, then you apply your pi half pulse, and then, you detect the NMR signal, and then, you start again polarizing and so on. But you see, on the long term, this Overhauser for bio application needs to be removed. And of course, just getting this factor of 200 back needs a lot of developments.
Yeah, and then because this relates also as to the, so you say cluster, I don't know, this KSR pass sequence.
Yeah.
So what limits exactly the line width of this?
Well, here really like our own limitations. So now I think I mentioned this quickly before. So when we did this experiment, so we had I bought like an objective, which was supposed to be non-magnetic by a special company, a lot of money. When you put it in, there was no NMR signal there.
No.
And what was the issue? They removed everything of this objective, but the coating was made of nickel. And this nickel coating, was still so magnetic that it killed our NMR signal. And then we were very lucky that we had like one objective around, randomly, which reduced it much more. But you see here the line width is also like 10 to 15 hertz. And we know if we move the optics and go back to our standard non-imaging experiment, we should be like 2 hertz. So at the moment we're limited by our engineering capabilities, unfortunately. Well, fortunately, but it's a hard work, to get rid of the line width. But it's not limited by the NV properties.
Yes, yeah. And then the last question, I mean, related to how long it takes. I know you said we use this 200 times in principle improvement of signal with Overhauser polarization. But let's say to get this image, I mean, how long it takes to get that image?
So we just had the proofs yesterday. So I think we wrote here like it's 14 hours, and this might be shocking now. Wait. And it's not so bad. So the 14 hours, we had like 3 seconds duty cycle, which is kind of it's realistic if you have non-Overhauser, but we could measure much faster because the temperature kind of kills your T1. But let's assume that's fine. But as I said before, we are at the moment limited massively by the noise of the camera. So we didn't have enough light because at the end, everything has to fit. You need enough light on your sensor, the NV layer thickness, and so on. So I think by adjusting what we're doing at the moment, like the sensor on the NV density, the light you have, should easily give you a 10, maybe even factor of 100 more sensitivity. So the main thing was here, you see, also we totally do oversampling, and the sensitivities here was like, a couple of tens of nanotesla per cubic micrometer. There are other groups, have shown much better numbers, but in particular, because here we really tried to get the NMR signal and totally neglected anything on the sensitivities. That's really what we're doing now. I'm pretty sure we can improve this in orders of magnitude. But again, it's still hard for biology, really to go like down to very, very low concentrations will be hard. But so what I'm saying, don't be shocked if you look at the paper and say like, oh, this 10 hours of averaging. This is really, we did not focus for this publication here on the sensitivity.
Yeah. I mean, it's very nice. Wide field is very interesting to many people. I mean, very nice.
Yeah, there's many things to tune. Yeah.
Yeah.
But it's still not, that we get the signal like, in a second. This is, of course, it's just NMR at the end.
Maybe I have another question, but maybe I leave maybe there are other questions from the audience, and we'll get back later.
Please.
Yeah.
So
the question I have related to this technique, because you said the change the A1 frequency, I think, to this time readout for the second part of talk or third part. So my question, you said you changed two frequencies. Like I understood. I think there is a schematic of the setup when you have. I think maybe next slide. The A1, when you show the A1 schematics. That one. So just to be sure here, so you are changing only the frequency of the laser, or you are changing the position? You are scanning the beam. I mean, you change the position. I'm just confused.
Right, no positions. What we do in this case, so if you only want to have two spots, what we did, you are from the field, we take like a double wind freak. So we take two frequencies, put a switch there, and feed these two frequencies into the AOM.
Okay.
But it's a frequency which goes to the AOM because, this basically depends, the angle of your deflection.
So basically you have two beams going to your sample. I'm just confused. Or
just like one. You have still one beam, but you basically feed two different frequency into the AOM. And then basically with this, basically you can very fast scanning.
So you think, and then you synchronize to your detection scheme.
Exactly. Yeah. You see, so look at the colors here. So one is like this red, one is kind of this violet one. So the first would be position 1, the second would be here. And of course, if you have only two frequencies, it's very easily doable by just taking one signal source with two outputs. If you want to have many frequencies, so many different spots, then you need something like an AWG because you need like the different frequencies to change very quickly. That's the point.
And you said here you need to what's the maximum time? I mean, readout time, because you said you need to correlate with microwave pulse the sequence. But usually,
so we typically have 5 microsecond laser pulses, and then in the time in between can be really, I think, can be like hundreds of nanoseconds. I think we have like a microsecond to be sure. But it's really I can really strongly recommend. It's very easy to implement.
Okay. Yeah, for sure. Maybe we'll get back to you. My students. Very nice.
Please also shoot an email because a second. Then be nice to meet you.
Okay, sounds good. Thank you very much. Very nice presentation. That's all.
Thank you. You're welcome.
Great. We can switch to next questions. Let me ask my question. So, Dominik, we all know the detection of metabolites at subcellular level would be extremely impactful for life science research and applications. So, at a large scale, what would be two most critical challenges from your perspective to overcome in this technology in order to be able to start to measure, say, micromolar level of metabolites inside live cells?
So at the
or
end,
extracellular, say.
Yeah. So at the end, it's the sensitivity. And I think, and what you just said, like, subcellular, micromolar, I would say, I would dare to say it's maybe impossible. Just, you do the math, if you have like a picoliter of your cell, times micromolar, there's only a few molecules left at the end. So even if you do, let's go back to the scale I had before. If you have like, the hyperpolarization, you can, for example, hyperpolarize your, oops, now that's stuck. Doesn't matter. So if let's assume you can hyperpolarize your sample, like, your metabolite, whatever your sugar you have in there, 100%, even then, I would say that you will always be like stuck on a cellular level, which I still think is very important. I think you don't need subcellular for many applications. But so what I would say is feasible is doing this like on a full cell volume, on maybe like a millimolar, maybe like a high micromolar concentration. This could be possible, but as said, only also with hyperpolarization techniques. Of course, then, the entire technique becomes very complicated because you need hyperpolarization, you need the NV, so the technology becomes very, very complicated. But then I would say it would be possible to do this that you get, per cell volume, basically a spectrum. This is possible, but subcellular, because you can think about, the volume also, of course, scales then, cubic. So cutting it down, like, from 10 micrometer to 1 micrometer, your signal, of course, gets weaker and weaker.
Yeah, and there are many micron size cells, or even 30
exactly. So I think, I think this 10 micrometer is a good length scale, but we have to be also at some point realistic here because, just a small volume with a low concentration at some point is getting really, really demanding. But also I want to emphasize.
And what if say nanodiamonds, which will be very, very close to analyte inside cells, right?
Exactly.
Their drawback is, of course, still T2 time, which needs to be improved, and we all try hard to
and I would say
then what you can.
The main issue here will be, of course, coherence time, but also our coherence time is not that good, to be honest, is the tumbling. Because, at the end, if you want to do this, let's say, more advanced quantum sensing schemes, you need to align a magnetic field with the NV axis. And that's the issue. So, but of course, could be one idea to put like a nanodiamond in the cell, fix it somewhere, align a magnetic field, and then do the stuff we do here. But also keep in mind then, we need a lot of laser power. So what I've heard, from nanodiamond people, they use like microwatts. We use like, hundreds of milliwatts. So at the moment, it's outside of the diamond because it's trapped in the diamond. If you would then have your nanodiamond in the cell, you would put in this laser power, into the cell. So also this comes with other issues. But let me just to the previous comment about the metabolism. So I really think also looking at water properties of cells is very, very important. And also here, of course, NMR is very unique. There's no other technique which can kind of, maybe infrared, but there's not that many techniques which can get this kind of really broad information of diffusion, of relaxation times of water. And this is, I think, also probably the first target we should aim for. Can we detect or can we analyze the water properties of single cells? But, but I have to disappoint you, I think really subcellular will be very complicated. But, also let's see how the developments go. And I fully agree, putting a nanodiamond there and maybe thinking about different ways to initialize, maybe less laser power could be feasible. But it's a long way.
And what about high magnetic field?
Right, this is of course the other question, right? If you would, increase your magnetic field, you typically increase your sensitivity. But for the NV center, that's exactly the technical challenge. So we're working on this now for a couple of years, is because if you increase the magnetic field, also your alarm frequency of your NV center will go up. So we typically work at this like, tens of a Tesla, you have 2 gigahertz. That's easy, that's very easy to handle. But if you go to 1 Tesla, you are at, 30 gigahertz. And of course, at the same scale, also your alarm frequency of the protons you detect goes up. So you have to pulse even faster your electronic spin. And I would say that, 1 Tesla is feasible. Going beyond, of course, there's some pulse sequences proposed to go higher here. But I would say 1 Tesla is feasible. After that, really, we have to see, where to go, and also from the complexity, how much sense it makes. But also we have to say there's benchtop NMR available these days, which work at 1 Tesla. So small molecules, for example, can be easily analyzed also at 1 Tesla or maybe 1.5 Tesla. So I think we don't need for all applications, it's very, very high fields. Of course, would be nice to have, but I think for especially specific applications, what the NV does, I think also like magnetic fields up to 1 Tesla could be very beneficial.
Thank you so much. Please ask your questions. Don't be shy.
Dominic, I got a quick question. Your microwave antenna, so maybe you touched on this, but so is your setup, would you consider it more like a resonator? Or, so does your microwave antenna have tunability, or is it like a large bandwidth kind of?
Right. Okay, that's always embarrassing. You saw basically, I think, the image that we still use this kind of shortened coax cable for this application here. So in this case, it was only this shortened coax cable, which works amazingly well. That's why we use it. And we have now also a new study where we use like resonators, and the resonators, of course, come with many other advantages, such as, so if you want to do the imaging on a large field of view, you need a very homogeneous field. This wire won't work. So then, of course, you have to go to resonators. But the resonators at the same time we've seen comes with many other things because they're narrow band, they shift and so on. So all what you have seen here on this presentation was all done with this broadband primitive short coax cable. We're using resonators. We have a new study on this on NPJ Quantum. We use resonators, but they come with a lot of other trouble, of course. So I think on the long term, it's absolutely the way to go with resonators, not for all applications, but for many. Also, in particular for the high fields. But in our case, my experience was it comes with a lot of technical overhead.
Mhm.
Felix, please.
Thank you. Yeah, the first is I was just wondering about what your spatial
I
range
think
is.
it's
It is very hard to hear. Can you raise your volume, please?
Is this better?
Yeah,
Oh,
much better.
much better.
Okay. Yeah, some really incredible techniques. I was just wondering, yeah, first, what is your spatial range with this AOM scanning approach before you kind of lose laser intensity and stuff?
Right. Yeah, very good point. So I'm also not a 100% expert here on the AOM. So we used to begin AOM, but there's also AOD. So an AOD is like a deflector, which is I think optimized what I was told for getting large deflections. So but we use the standard AOM and we people using, and I think it's a 250 megahertz device, and I think we can do plus minus 40 megahertz. And then of course you see if we go beyond that, then the deflection efficiency goes down and the intensity goes down. But this is still, you see, also in this case, we could scan, I think now maybe like a couple of hundreds of micrometer, maybe 200-300 micrometer you could scan. But as I said, so we have optics students on this, and he always complains that, Dominik, we have to buy the AODs, which are made for that. Right, but I think the beautiness is that basically all the NV experiments have it intrinsically there. Therefore, I think the technique is so nice. I think if you also want to do, of course, 2D scanning, which of course is the obvious idea at the end, then I think one should change maybe to one of these, AODs, which are commercially available exactly for these purposes.
Cool. And the other thing I was wondering about is, what do you think your prospects of this wide field NMR are towards pushing towards chemical shift resolution
Absolutely.
through
So that's
dissipative decoupling, or I know there's some repetitive readout demonstrations with these ensembles. There's a paper out there. You mentioned you were limited by your camera and electronics, but maybe you kind of already touched on those things, but
you mean this for sensitivity improvement, or?
Yeah, kind of to get sensitivity improvement and also frequency resolution improvement.
Right, yeah. Well, so I think in the current case, in principle, you could just put, any sample in there. You would get basically, if you were to take a standard sample with 2 lines, you would see that. Why we didn't do that, it was at the moment I would like to avoid that people say again, you get these, shitty, spectra at low field. So I think what we really have to change here would be go to higher field. And you see then all of the issues come in that you need then you cannot work with a loop anymore, you need a resonator and so on. But this is exactly what I think also the strength apart from the bio application will be, because you can do this highly parallelized NMR. Totally before we have 1 million spectra, of course, typically we will have only then, maybe thousands of spectra. But this is, I think, the powerfulness of this technique. And as you just said, even now we would get a spectra, but it's not nice because we do it at low field. So therefore, I think we have to go to half a Tesla at least to get this chemical, shift resolved resolution to get spectra here. This is ongoing work. But even now, so this is no limitation at the moment. So if you would put some sample in there which has a chemical shift at this field or J coupling, you would see that. This is not limited by that at the moment.
Okay. Yeah, I thought maybe the NV density, maybe you have lower T2 stars and so on.
No. So then we're not limited by that. Yeah. So it's really at the moment, it's only by basically the magnetic field we're working at. Yeah. So if you would have a sample with a large chemical shift, at this field, you could put it in and we would see it instantaneously.
Fantastic.
But again, it's also a sensitivity thing, and so on. So that's, it's that really something useful. That's what we are working on. It's, it's not nothing you do overnight. That's what I'm saying. So I think it must be a combination of, going to higher fields, getting a good resonator, just the previous question, and so on. So there's a lot of homework to be done to get really to a useful technique at the end.
And so was your camera like pulsable at nanosecond kind of timescale resolution to gain these spectra?
So the camera, it was one of the main works. So, we use like a streaming camera because, at the end you want to average. So there's many high-speed cameras available, but the issue is always, they fill up the internal memory and then basically you have to read out this over like 20 minutes. And this is not what we want to have because you want to, we want to average over time. So we have a streaming camera. So streaming basically means, you can read out the data all the time. And the streaming camera had like a maximum frame rate, I think, from 3,500 frames per second. And then if you reduce your field of view, you can go up to 6,000. So most experiments were at 6,000 frames per second. This still means we are much slower than our actually pulse sequence. Our pulse sequence typically, I think, 50, 60 microseconds long. So that's the next point. You see, we are kind of throwing out a lot of like data, or like we don't record data. We could have recorded data. So ideally, would to have like a camera with 10,000 frames per second. Again, on free streaming, but at some point, one of the issues that you get tons of data, of course, which you also have to digest.
Yeah.
And then you may ask, there's like this famous Heliotis camera around, this lock-in camera many people using in the V community. Also, we use that at the beginning, but exactly this issue that it can do very fast acquisition of data, but then it's limited how many really frames you can get out per second, and also it has this dead time of 10 seconds. For that reason, at some point, we went to these kind of streaming cameras, which we think for our applications are the best. But again, we have, what I've mentioned before, so this 10 hours of averaging, which is shocking, is really at the moment a limitation of our technology, of the equipment we have, So I think, if you find the right camera, if you have enough light, then, these things can be easily improved orders of magnitude.
Yeah.
But, it's a streaming camera.
So you kind of, you go back to the Z-axis and then you kind of have to wait. And during that time, T1 is happening, and then you can take your acquisition, and then it sort of slows down your timing resolution, or I guess
so we do this kind of Kaiser protocol where you kind of have like this dynamical decoupling sequence which you repeat, and this locks in basically to NMR signal. But at the moment, this pulse sequence is 50 microseconds long, but then you have to wait, until the next frame of the camera comes. You wait, whatever, 200 microseconds or 150 microseconds. So ideally, so if the pulse sequence you see would have something like 20 kilohertz readout, because the pulse sequence is 50 microseconds long. But 20 kilohertz on a streaming camera, on larger field of view, it's complicated.
Cool. Yeah, thank you.
But it's a very fast development of cameras, so I think we can be optimistic here. But of course, you may also just choose a smaller area of your camera, because then you increase the speed, of course.
Yeah, cheers.
Yeah, also, if there's more questions, just shoot an email.
That would be great.
Thank you so much to our audience for great questions. I can see Dominik really enjoyed them, right? So one last question, if somebody else has one. If not, then I would like, on behalf of everybody here, to thank Dominik for his very exciting presentation, excellent presentation, and we all wish you great achievements in NV-based NMR technologies development in 2025.
Thank you very much. Great work. Very nice work.
Yeah, please contact me just if there's more questions. I'm happy to have a second Zoom.
Okay. Yeah, and Ashoka Joy just committed that it's great work. So he echoes me.
Okay, have a nice day. Bye.